Ariana, Zoom is doing good and volume is okay. Well, I want to welcome our seminar presenter for today, Jen Murphy, is joining us from University of Toronto. I'll start by saying Jen has been a great colleague and friend for over 20 years now.
Yeah.
Say that out loud. So I have a background on Jen here. I'm just gonna read through quickly, a little more information about her current and past experience. So Jen Murphy is professor at the Department of Chemistry at the University of Toronto, where she teaches and mentors a team of researchers who focus mainly on understanding the sources and fates of atmospheric pollutants and greenhouse gases. Her team has performed fieldwork across Ontario and California, Utah, Colorado, northern Michigan, the Alberta oil sands region, and Canadian Arctic. Well, Nova Scotia.
Yes.
They deploy scientific instruments at ground sites, on tall towers, stratospheric balloons, aircraft, and ships to measure chemicals in the atmosphere. So just about every platform you can think of. In addition, they leverage government monitoring data sets to characterize spatial and temporal trends in air pollution and the roles of emissions and meteorology. She has served as a member of the Scientific Steering Committee for the International Global Atmospheric Chemistry, or IGAC, project from 2016 to 2020. Has been an associate editor for the Journal of Geophysical Research Atmospheres since 2020. In 2019, she was a recipient of the American Geophysical Union Ascent Award for Mid-Career Excellence in Atmospheric Science. In 2020, she was a member of the team who won the Brockhouse Canada Prize for Interdisciplinary Research in Science and Engineering. And in 2024, she received the Canadian Society of Chemistry's Clara Benson Award. Jennifer completed her bachelor's at McGill University in chemistry with a minor in environmental studies. During her PhD in chemistry at the University of California in Berkeley, she tracked the sources of air pollution influencing the deposition of nutrients in nutrients to Lake Tahoe, and studied smog in Central California, and met some of the coolest people that she's ever met during that time. As a postdoc fellow at the University of East Anglia in 2006, she operated a mass spectrometer aboard a research aircraft flying over West Africa to measure volatile organic compounds emitted from tropical forests. She also has a lot of really interesting stories about that, about doing field work in West Africa. So today she's going to talk to us about surface atmosphere exchange of ammonia across ecosystems.
Great, thanks, Brent. So it's really nice to be here, and I'm really enjoying this beautiful snowy scene out the window, and hoping my flight will be able to fly tomorrow. But while I'm here, I'm really excited to share some of the research we've been doing in my group. So what I want to focus on today are some stories about our measurements of surface atmosphere exchange across different ecosystems. So I'm going to share some stories from these different environments, and I'll get more into them in a moment. But yeah, so an agricultural environment, a remote Arctic environment, and then a sort of urban environment. So I'm going to be sharing work that spans many years of the group, and so these are a few group photos from different periods of the group. I've been really lucky to work with a nice cohort of students and postdocs during my time in Toronto, and we've had funding and collaborations from several different agencies that have helped support this work. So as I mentioned, I'm going to talk about surface atmosphere exchange. So the first story I'm going to tell you about is work we did in an agricultural region looking at exchange of ammonia between a farm that had been cultivated to grow corn over 2 growing seasons. And then I'll tell you about some work we did in the Canadian Arctic archipelago. And then I'll finish up with some work we've been doing more recently closer to home. So I'm going to tell some stories about the sort of fate of ammonia in the atmosphere. And so I used this infographic in my talk in chemistry yesterday as well. This is sort of how I've been thinking about the behavior of this molecule ammonia in the environment. So it's emitted from several sources. So globally, agriculture is the dominant source. But depending on the location you're in, things like point sources or vehicles or fires can also be important. And a lot of our motivation for studying ammonia is because it plays an important role in the formation of fine particulate matter, and also the phase partitioning of ammonia is really rapid and it can be really variable. And so if we want to understand what's going on with ammonia, it is very helpful to be making measurements of ammonium at the same time. And so sometimes I might refer to this quantity NHX, which is the sum of the gas and particle phase. I talked a little bit yesterday about the fact that ammonia doesn't oxidize very fast. Its lifetime against oxidation is about a month. But this is a small sink of ammonia, but potentially a source of N2O that's growing in importance. But mostly what happens to NHX is that it deposits either through wet or dry deposition, and that can potentially lead to changes in the nitrogen status of an ecosystem, and in extreme cases can lead to eutrophication or things like that. But what I'm going to focus on mostly today is the challenging part of, one of the more challenging parts of understanding ammonia's budget is the possibility that it can undergo bidirectional exchange. And so we can have deposition of ammonia to an unmanaged ecosystem or an impervious surface or body of water. And if the conditions change, then there may be the potential to re-emit that ammonia again. And so when we're trying to understand budgets of ammonia, this is a complicated thing, because we can be misrepresenting it as sort of a gross emission or gross deposition flux, and then we could end up kind of double counting it or misunderstanding how to keep track of how much new ammonia is sort of entering this environmental compartment. So since this idea of bidirectional flux is so important for the work that we do, I'm going to take a moment to sort of explain the framework that we use to understand it. So imagine we have a solution here, an aqueous solution. So this could be the ocean, it could be a cloud droplet, it could be the water in the soil pore space. So there's some amount of ammonium there, and that ammonium ion is in equilibrium with neutral ammonia, and that equilibrium is governed by the acid dissociation constant and depends, of course, on the amount of protons. And then that neutral aqueous ammonia is also in equilibrium with gas phase ammonia, and that's regulated by its Henry's law constant. And we can solve for this quantity chi. This is basically the gas phase concentration that's in equilibrium with a given amount of ammonium and H plus concentration. So this, we sometimes use gamma for the emission potential, which is the ratio of ammonium to H plus. So if you have more ammonium relative to H plus, that means you're going to have more ammonia in the gas phase. And then these numbers are just sort of canceling units. And this piece in the temperature exponent reflects the enthalpy of solubility and the enthalpy of acid dissociation. And they're both strongly temperature dependent. And so this ends up the amount of ammonia you have in the gas phase for a certain solution composition is extremely temperature dependent. So we expect a lot more ammonia in the air over a solution when the temperature is higher. So this is what thermodynamics tells us should be in the air when that air is in contact and equilibrated with a solution. But of course, there's other things going on. And so the actual concentration of ammonia might be different than that equilibrated amount. And so the idea is that this actual concentration is always tending towards this equilibrium amount, but there's some mass transfer resistance that's stopping it from immediately equilibrating with that. And so we have a flux between the atmosphere and the surface that's driven by the magnitude of the difference between the actual concentration and the theoretically equilibrated concentration, and it's modified by that exchange velocity. So this is this idea has existed for over 40 years, and it's been used for many decades to sort of try and characterize different ecosystem surfaces and the ocean, for example, and predict whether that reservoir should be a source of ammonia or a sink of ammonia. So I'm going to start off with a story about the study we did on a experimental farm. And so this was in collaboration with folks at the Agriculture and Agri-Foods Canada. And they were interested in understanding emissions, that sort of loss of nitrogen when it's applied as a fertilizer. And some of the motivation for that comes from a figure from this paper showing that when nitrogen is applied in the form of urea, a pretty substantial fraction of the nitrogen in the urea is lost to the atmosphere as ammonia, on the order of 10 to 30%, which is sort of a waste of resources for the farmer themselves, but also can create significant air quality issues in the region. So these were data from a decade ago, sort of looking at a global meta-analysis. And then we were planning to work in a field where corn was being grown. And there were a couple of studies that were done in North America over cornfields, or at least fields that were cultivated with corn. Of course, it almost doesn't matter what the crop is when the fertilizer is applied, because it's usually applied around the same time that the seeds are planted. So the crop itself is sort of in the early period, not that important. And in those studies, several percent of the nitrogen was lost as ammonia, though not as high as this sort of meta-analysis. And these studies, it was a combination of urea and ammonium nitrate. So urea takes a while to mineralize to form ammonia. So that's one of the advantages of applying urea, is that it's possible, it's thought that if you add the nitrogen in a form that's not already ammonia, that it will start to become available as ammonia on the sort of time scale that the plant is closer to when the plant is like accumulating biomass and can use it rather than having it leave the system. So this is a overview of the site where we performed the research. So this is a field that is operated by Agriculture and Agri-Food Canada, but they actually hire a local producer to kind of cultivate the land in a manner that's sort of relevant to the local context. There's a large trailer here where some instrumentation is located, including an instrument to measure N2O, for example. But when it comes to measuring ammonia fluxes, because ammonia is so sticky, we need to have our sensor right out in the field, very close to where the fluxes are being measured. So we located, so here there's these two flux towers, and the idea is that there are different treatments on this field, and regardless of the wind direction, each flux tower will be measuring one of the treatments, and our collaborators were using that to understand whether applying inhibitors, nitrification inhibitors, was impactful on the N2O fluxes. Since we only have one instrument and we have to be right at the tower, we just located our instrument here. So the instrument is inside this sort of coffin that is very well temperature insulated. And we have quite a short inlet. And so we're sampling the ammonia very close to where we're making the wind measurements for our eddy covariance observations. And so the fertilizer was applied at a rate of about 150 kilograms of nitrogen per hectare. I'm not going to talk about the impact of these inhibitors because we didn't have very much visibility onto that, but that was another part of the project. So just to give you a taste of what some of the data looked like. So this project was led by a former postdoc, Alex Moravec, and in the second year we were there, another postdoc, Soumya Singh, contributed as well. And so I know there's some folks in the room who do eddy covariance, and so you know that what we're trying to do is look at the covariance between the vertical wind and the compound that we're trying to measure. And so I think you can see that there is some information. You can see that the ammonia, the variability in the ammonia at high time resolution has some similarity to the variability in the wind, as does the variability in temperature. And so one of the big challenges to try and do this work was that getting high resolution ammonia data is really challenging because of all the sampling issues that we can have. So the signal can really smear out as we're trying to bring it into our instrument. And so that was sort of the first big thing that Alex Moravec worked on. So if I direct your attention to this left-hand panel first, what I'm showing you is some work done by the first PhD student in the group. So what we did was introduce a sort of square wave amount of ammonia into the instrument and look to see how well we could capture, you know, a high concentration and then an immediate transition to effectively zero. And we regularly see this exponential decay, and we can usually characterize it by sort of a two-factor exponential decay. So the first time constant represents approximately the amount of time it takes to just turn the air over in the optical cell itself. And then there's a second time constant that represents what it takes to sort of pull all the ammonia off of all the surfaces that are upstream of the inlet. And that time constant can be a lot longer and can be problematic if you're trying to measure high frequency fluxes. And so we came up, Raluca in her work came up with this factor D, which was represented what proportion of the response is slow. And so we find that that depends on things like the humidity and the absolute concentration as well. So the first thing that Alex did when he was trying to characterize the fluxes was look to see how the proportion of the true flux, you know, how much of the actual flux do we measure as a function of D. So when D is 0, that means the time response is dominated by the fast time response, which is good. So we measure almost all of the flux. And as D becomes bigger, so as more and more of the time response of the system gets driven by the slow response, we see less and less of the flux. So he did this very careful characterization that also depends on the wind speed. And the upshot was that we needed to correct our flux measurements by between 20 and 60%. So it's not a perfect technique, but we characterized it pretty well. And, you know, we've applied that correction. And of course, there's uncertainties that go along with that, but we think we understand pretty well how to interpret the data. So now I'm showing you a time series of the ammonia flux, starting in late May and running through the entire growing season to the end of October. And so the urea in this growing season, it was applied on May 25th, and the corn was harvested. So this was silage corn. It was harvested pretty late in November. And so if you look at the fluxes, you see there's sort of this initial period of emissions, and then there's a period in June of mostly deposition. And then in the sort of all throughout August, we saw lots of emissions. And then in the fall, we saw a lot of net deposition. So if we focus on this early time period, what surprised us was that less than about 0.1% of the fertilizer nitrogen that was applied was lost as ammonia. So this was a lot less than we were expecting. Of course, this is field work and things don't always go the way you want. And they ended up applying the fertilizer before we were ready to make the measurements. And so we actually missed the first 2 days post fertilizer application. And so that was a bummer because of course, when our measurements started, they were like really high and then, you know, quickly went down. And so we're sort of blind to what happened in this period. And so it's possible that they were way higher for longer. We have some offline flux measurements by relaxed eddy accumulation that tell us that that's probably not true, but this was a sort of a bit of a gap, obviously, in the analysis. So when we went back out in 2018, we set up earlier at the very beginning of May, and the fertilizer was applied. You know, we had 2 weeks of data before the fertilizer was applied, but as you can see, there were not very significant emissions after the fertilizer was applied. And so, yeah, that was, we were like, okay, well, maybe we didn't miss much this time, but we still don't know what it was. Interestingly, I just zoomed in a little bit more closely on this y-axis here. And often what folks see is that the ammonia doesn't start being emitted from urea fertilized fields until after the first rainfall, because the rain is needed to wake up the microbes whose enzymes are being used to do this hydrolysis. And so if you look right after this rainfall, you will see a pattern of like nice diurnal fluxes for the 4 days following the rainfall, but the magnitude is really, really tiny. Like these fluxes are minuscule. So this shows we can measure small fluxes reliably, but the surprise is still that they don't match what we were expecting from other reports of ammonia emissions from this kind of activity. So so we looked in more detail, and again, if we, a lot of other researchers have reported the cumulative fluxes in the first 3 weeks following fertilizer application, and as I said, it's a really tiny fraction. So just for context, if we had measured 4 or 5% like these researchers had, what would that look like here? It would look like sort of fluxes of this magnitude for that long. So it's really different, and we're really sure that we're not just not measuring it properly, that's definitely not what's happening. So there's something different about our site. So Alex went back to try and use this bidirectional framework to see if we could understand what fluxes we should have expected based on what's going on in the soil. So one of our regrets is that we only made sporadic measurements of the ammonium content and pH of the soil pre and post fertilization. And the soil is very heterogeneous. So we collected lots and lots of samples, but we only did that every few days. And so the range of emission potentials we got from those observations are pretty large. But what we were able to do is calculate a sort of continuous time series of this compensation point that we would have expected. And then we have our actual ammonia measurements, and we have information about the wind. And so using this model, we were able to predict in the red bars the magnitude of the fluxes we would have expected to see based on the chemistry of the soil and the ambient air concentration. And then in blue is the magnitude of the fluxes we actually observed. So there's that high skew there in the first year. And so these don't agree super well, but they're like a factor of 2 or 4 different. They're not like orders of magnitude different. So we interpret this to mean that within our ability to know what this emission potential from the soil is, our flux measurements are pretty consistent with that emission potential. So maybe then the question is like, why is the emission potential so low, or why is the compensation point so low? So I'll come back to that in a minute, but I want to talk about our attempts to understand the later part of the growing season when the canopy is more developed. And so Eiko Nemetz has published a framework kind of looking at this two-layer compensation point model. So considering not just the ammonium in the soil as a potential source, but also exchange between the air and the plants themselves via their stomata. And so Alex built a model where we have to assign the plants themselves an emission potential. And so we got that from the literature. It's a very specific number for a very uncertain quantity. And then because we made these periodic measurements of the soil emission potential ourselves, we could use the values from our own measurements to constrain that model. So Alex built this model. He sort of ran a forward version of this model to simulate what we expect the emissions to be across the growing season. And so what I'm showing you here are, again, our measurements in the blue and our model in the red. And I was actually really surprised, or pleasantly surprised, like the model does a good job a lot of the time. So during this deposition period, during this emission period, and during this deposition period, the model is simulating, approximately the right sign and magnitude of the fluxes. But of course, there's a period in the middle where it's not doing a very good job. And so what Alex has been working on is trying to sort of invert the model and try and solve for the uncertainties. And for example, you can see this is the attribution of the fluxes in the model to the ground or the stomata. And so during this period where we're not doing a very good job, it's where the plants sort of have fully developed. They've actually cut off the sort of physical exchange of air between the ground and above the canopy. And so according to the model, they're really dominating the fluxes, but the model doesn't seem to be doing a very good job there. So we're still trying to understand how to invert, how to use our observations as sort of a top-down constraint to sort of partition the fluxes between the components in the model. So what we've learned from this study is the emissions from ammonia can be small. We think part of the reason that the fluxes were so small in our study is that the method of application of the soil was that, there was some incorporation of the nitrogen into the soil below the surface. The conversion of urea to ammonia seemed to be slow in our system. So we didn't see very much of the nitrogen that was applied as urea showing up as ammonia in our soil measurements. And I think a specific thing that was relevant in our study that might be also relevant in a place like Minnesota is the soil temperature is really low. Like at the beginning of May, the soil might be like 5 Celsius or something like that. And so because of that exponential temperature dependence, that seems to be, a contributing factor. If we made the soil temperature 20 Celsius, the model would have way, way bigger emissions. So that could be part of it. So the sort of timescale of the urea turning into ammonia, the competition for the ammonia through this nitrification pathway, and then the sort of possibility of these emissions, these factors are all competing, but we don't totally understand how. We also found that the emissions of ammonia in the later part of the growing season from the canopy might actually be larger than the emissions from the soil. And we're still struggling to partition those fluxes to the plants. So they could be coming out of the leaves of the plants through the stomata, but there also could be a sort of reservoir on the cuticles of the plant leaves, and we're struggling to differentiate those sources. Okay, so that's a view from the farm. So now I'm going to take you to the Arctic. And so this research was motivated by a desire to better understand the climate in the Arctic. And in particular, during the winter in the Arctic, there's often this sort of haze layer. I don't know if you can make it out, this sort of gray layer. And whereas in the summer over the Arctic, the air is really clean. And the reason for that is that the transport patterns that prevail in the winter, the Arctic front is at much lower latitudes, which means that air from the sort of northern mid-latitudes can get into the Arctic, and a lot of long-range transport can bring dirty air into the high Arctic. But in the summer, the Arctic front contracts. And so if you are in this zone, you only are being influenced by very local sources, and there's not a lot of emission sources in the high Arctic. And so the air tends to be much cleaner. And so there's a couple of measurement sites in the high Arctic here at Alert and here at Zeppelin. And if you look at the number distribution as a function of particle diameter, you can see that in the winter months, there's lots of particles that are, several hundred nanometers in diameter, the sort of long-range transported pollution. But when we enter into the summer months, the size distribution of particles tends to be dominated by these much smaller particles. And we think that they are not being transported to the Arctic, but they're being generated through new particle formation. And they can be very important for climate as we want to understand better what's going on. So that was the motivation for this large field campaign that was led by my colleague, John Abbott. And so we went to the Arctic and tried to understand the interplay between emissions of gases from the ocean and primary emissions of particles from the ocean, the impact of these different particles on forming clouds and their climate impact, and then the importance potentially of transport, including from fires. And there's a non-zero chance, let's say, that we're going to start seeing lots of shipping and maybe resource extraction and tourism in the Arctic. And so what impact might new local anthropogenic pollution sources have in the Arctic? So our ammonia bidirectional exchange framework kind of fits into this motif. We have dimethyl sulfide is emitted by phytoplankton. It can go into the atmosphere. It's pretty volatile, so it escapes the ocean into the atmosphere. It gets oxidized to form these low volatility sulfur gases, which are known to nucleate new particles. But that rate of new particle formation is much faster in the presence of ammonia. And if there's ammonium in the ocean, then there should be ammonia present in the air. And there's sort of this longstanding observation that the ratio of ammonium to sulfate in the particles in the atmosphere in remote regions is pretty constant. And originally, people thought that maybe it has something to do with the coupling of the sulfur and nitrogen biogeochemical cycles in the ocean. But actually, if you have bidirectional exchange of ammonia, you can imagine that if more sulfur is coming out of the ocean and making these acidic particles, that would actually change the direction of the ammonia emissions as well. So that could be like forcing this coupling to happen. So people have measured the bidirectional flux of ammonia between the atmosphere and the ocean on several cruises. So this is a sort of summary of what was known a couple of decades ago. So some papers find emissions from the ocean, some papers find fluxes of both directions and with some deposition. And in this paper by Martin Johnson, he kind of organized all the observations that had been made from research cruises. And he showed as you go further north, so along the x-axis, so the gray line here is a flux of 0. And so in the high latitudes, the fluxes were almost always deposition into the ocean. So I was excited about the chance to go to the Arctic, in part because around the time we were putting our plans together, Julia Schmala published a paper where they made measurements in the Southern Ocean. And what she saw was a lot of organic nitrogen and ammonium when they were making measurements near, I think it's actually called Bird Island. So it's pretty obvious what the source was. There's a lot of seabird colonies on islands in the Southern Ocean, and all birds excrete their waste predominantly as uric acid. And when uric acid mineralizes, just like urea, you're starting off with a bigger molecule, but eventually a lot of it turns into ammonia. So Stuart Riddick, who I believe did this work at CEH, developed a sort of compilation of likely emissions of ammonia from seabirds. And you can see that the part of the Arctic that we were heading towards had some of the bigger sources. So we wondered if we would see that. So it was really a privilege to get the chance to spend some time in the Arctic. These are the crews that were on the ice-breaking ships when my team was on there. This is a view from the ship looking at the Polar 5 aircraft flying by us. So we use the ship as a research platform, but also as a source of black carbon for the aircraft to sample. And I'll also show you some measurements from the ground site at Alert as well. So the first deployment our team was part of, we went out on this icebreaker and we sailed, we left Quebec City and we sailed up the coast of Baffin Island and actually made a left turn here. The oceanographers swamped our inlet with ocean water, so it took us a while. We didn't have a lot of data for a while. And then we cleaned ourselves up and started seeing really high concentrations of ammonia in this part of the journey. And these concentrations are, up to 1 part per billion, which is not very significant for this part of the world, but there's no agriculture up there. There's very little fuel combustion other than our ship. And so we were kind of surprised to see such high ammonia levels. So we were able to collaborate with oceanographers to sample the ocean water and also melt ponds that form on the ice. So these melt ponds are basically mostly freshwater. So their pH is a lot lower and they're a little bit colder than the open ocean. And 3 of them had higher ammonium concentrations, but because they're more acidic and a little bit colder, the compensation point of the melt ponds is actually lower than the open ocean. But in both cases, these compensation points are, an order of magnitude or more lower than what we actually observed in the atmosphere. So this tells us that the ocean can't be a source. It's too cold or there's not enough nitrogen. And so the high levels of ammonia we saw couldn't be a consequence of outgassing from the ocean. So as we suspected, the birds seem to explain a lot of the high levels of ammonia we saw. So this is a map of the colonies that were in the Riddick inventory, and then we added in stars a few more colonies that we found out about from other sources. And then we worked with Betty Croft, who is in Randall Martin's group. She implemented those seabird emissions into the Geoschem model. And this figure is showing you the percent change in surface atmosphere ammonia concentrations because of the seabirds. And this is the part of the world we were sailing around in. And you can see that we went to the right place to see this impact. It was like a very large impact. There was almost no ammonia in the air if she left the birds out of the model, and there was a relatively large amount if the birds were included. So the aerosol scientists were really excited about this because prior to that, they hadn't been able to simulate these bursts of new particles that were being observed in the high Arctic. So this is a time series from ALERT in 2011. The black line is the number of particles with diameter less than 10 nanometers. And you can see this is going from April, May, June, sort of through the summer. And you can see that once June starts, there's these periodic bursts of large numbers of small particles. And in the model that did not have the seabirds, there's basically no ability in the model to generate new particles because the particle nucleation scheme needs ammonia. Then when she incorporated the seabirds, there was more ammonia, and you can see in the blue, it's not matching the episodes perfectly, but you're starting to see the same kind of thing. So all of a sudden the model is able to have these new particle formation events as a consequence of this ammonia. So she was able to go a step further and look at how do these small particles impact the cloud condensation nuclei, and for that we needed another piece of the NetCare campaign, which was to learn that there's lots of organic acids that are condensable, and those help these particles grow from 10 nanometers up to a couple hundred nanometers, where they can form a cloud condensation nuclei. So Betty was able to simulate the change in the cloud droplet number concentration as a result of the seabirds. So you can see that was a big change there. And that actually leads to a fairly substantial aerosol indirect effect of about 2 watts per meter squared in that very local region, specifically from the ammonia from the birds. So this was a fun paper to work on, and they did a great job, Betty and my student Greg, talking to the press, because this was sort of like the sacred and the profane, like climate change and bird poop. So I did one drive time radio show where there was like a lot of weird sound effects, and that was enough media coverage for me, but it was fun to work on this project. So the story, of course, wasn't totally that simple. We were impacted a lot by birds during during our travels through the Arctic. So in these maps, the blue is showing you where fires are, and the pink is showing you where the bird colonies are. So here's a time period where our observations, and then this color is telling you, where were we sensitive to emissions from? So during this period, pretty definitively, we were being impacted by the birds, but there were other periods where we saw high ammonia concentrations. And in that case, this emission sensitivity points us back towards the fires. So this was a very high fire season. So that was also part of the story. So we were fortunate to be able to go back in 2016. This time we put our higher time resolution instrument on the ship. And so in this map, the blue line is showing you where the ship went in 2014, and then the colored and gray symbols are telling you where the ship went in 2016. So we definitely spent time seeing high concentrations of ammonia, so kind of similar range of concentrations. But we also spent time in places where the ammonia had been high in previous years, and it was below our detection limit for a long time. So the story was a little bit different, but because we had really high time resolution with this instrument, we were able to see clear evidence of the impact of the bird colonies. So here's an example when the ship was passing by. So the colored dots here are large nesting sites of like millions of seabirds. And the orange area tells you where this sort of back trajectories tells us we were sensitive to. And if you look at the time series, this is like the raw ammonia data, just to show you how unambiguous it is. When we were passing by these bird colonies, we saw these really, really large enhancements in the ammonia. So definitely, when we get close to a bird colony, it's pretty obvious that the birds have a big impact. But in 2016, Greg took the instrument that he'd operated on the ship in 2014, and he deployed it at Alert, which is on the northern edge of Ellesmere Island. And he recorded this time series of ammonia concentrations. And we were, we really didn't know what to expect. We're not very close to bird colonies in this location, but it was a very warm summer, even by Arctic standards, even actually by Minnesota and Toronto standards, it was pretty warm. And so we were interested in sort of this pattern that we saw, and Greg collected a few samples of soil from the tundra, and he characterized the emission potential, and then he was able to simulate the compensation point from those soil samples. So he collected, triplicate samples at 3 locations. And when we used the, I think we used the air temperature in this case, we looked at the compensation point from those 3 samples. We saw, one of them had a really low emission potential. So that's the green soil here. That would say that concentration, that, sort of theoretical amount of ammonia over that soil was always well below what was observed. But the other 2 soil samples had actually high enough ammonia emission potentials, and the temperature, what's really happening, what's the difference between here and here is really the temperature. And you can see that the compensation point gets higher than the concentration, and also the concentration looks a lot like, the sort of variability. And so this to us is pretty strong evidence that the local soils could actually be a source of ammonia to the atmosphere in that particular context. And so if we look at the distribution of ammonia in the air from these different campaigns, I'm just showing you sort of a histogram. So when we went in 2014, we saw a median concentration of about 220 parts per trillion. That was actually perhaps randomly similar to what we measured with the same instrument in 2016. And on the ship, we made a lot more very low concentration measurements, but the average values are pretty similar. And again, 2014 was a year with lots and lots of fires in the Northwest Territories, whereas 2016 was one of the lowest fire years. And so I think fires definitely played a role here, but a high fire year and a low fire year, we were sailing around here. Those aren't too different. And then I was also able to find a data repository of observations from Zeppelin, which is on the other side of Greenland. And if I artistically show you their distribution of concentrations, it's actually remarkably similar. So I think that we have a reasonably good sort of sense of the range of ammonia concentrations in at least this particular sector of the Arctic. We were also able to collect wet deposition samples during these campaigns, and we could calculate the sort of net deposited NHX during these periods. And interestingly, a colleague analyzed the snow that fell at Alert over this 6-month time period, and we see, more nitrogen being deposited in 1 month of summer precipitation than in 6 months of winter precipitation. So even though there's a lot of pollution coming to the Arctic from the lower latitudes, when it comes to the nitrogen in this reduced form, the birds and or the tundra are, really important in the summer. And so I'm really interested in maybe going back to the Arctic and learning more about the tundra. So what Betty did, this was sort of a summary paper for this Arctic work, is she incorporated our emissions of ammonia from the tundra which she did by assuming the entire Arctic had the emission potential of one of our measurements. This is obviously not right. It might be approximately right, but who knows? But what she found is that she could, so as I told you before, the original model had a huge bias in particle numbers, which was improved when she added birds to the model, but she could get that sort of same type of improvement by incorporating this very simple representation of emissions from the tundra. And this source will be very sensitive to climate change. So we learned that, I didn't really show you these measurements, but almost all of the ammonia in the air is in the form of gas phase ammonia, not particle phase ammonium. The fires can matter some of the time. The seabird colonies can be important locally. We know there's definitely net depletion over the ocean, and over terrestrial ecosystems, we, just starting to scratch the surface of understanding. But it seems like bidirectional exchange could be important. So I've vastly overestimated how much time I have to tell you about things. So I'm just going to tell you one quick piece of the Toronto story. And that is related to this emission potential and our ability to actually take bulk soil samples and say something about how much ammonia should be in the air. And the piece that's missing in this story is that, of course, in soil pore water, the ammonium is not just going to be present, dissolved in the water, freely soluble, but some of the ammonium is also going to be adsorbed to the soil particles themselves. I'm by no means an expert in soil chemistry measurements. And so we sort of looked through the literature and looked at what techniques people used to quantify ammonium in soil. And we used a sort of standard exhaustive extraction technique, a very salty solution to sort of liberate all the ammonium from the soil. But what we actually want, what's actually relevant to the atmospheric equilibrium is just the sort of freely soluble nitrogen. And to make matters even worse, Matthew doesn't make matters worse. That's how Matthew. To make matters better, Matthew decided to tackle this. And so, again, sort of surveying the literature, being a bit naive, we were following the sort of standard methodology to use salt to extract for ammonium, but to use a deionized water slurry to measure the pH. And so those are influencing those ions in different ways. And so this ratio may not be exactly correct. So Matthew did a lot of work with different soil samples from around the city, and he measured the isotherms, and he measured them for, he saw that they were different depending on the cation exchange capacity of the soils. He found that a Temkin model with just 2 free parameters did a pretty good job of representing the sort of free amount of ammonium in solution relative to the total amount in the system. And even better, one of those parameters we can model using the cation exchange capacity, which is often a quantity we know about the soil. And so what he found is that the emission potential we get depends really strongly on the soil analysis protocol that we follow. So the standard way of doing things for us had been an approach that would yield these red points for the emission potential. If we follow this Temkin protocol, we get, often an order of magnitude lower values. Interestingly, he tried just extracting, he tried doing the pH measurement and the ammonia measurement with salt, and he tried doing them both with DI water. And those are actually closer to what we think is probably the right answer. So this is also obviously important because if your emission potential changes by a factor of 10, then your compensation point would change by a factor of 10, and that could change not just the magnitude, but even the direction of the flux that you infer. And so I would really like to go back and study more soils in the Arctic and do it in a more sort of thoughtful and nuanced way and see if it's really true that, there are soils across the tundra that could be such an important source. Okay, so I will leave it there for now and happy to take any questions.
You can start in the room with questions and you can let us know if there's some on the a question about your bird colonies.
Mhm.
It's so fascinating. At any point, was anybody measuring amines at all? Or have you on any of the?
Yeah, that's a good question. The system that we were running at Alert, well, actually, I mean, on the ship as well, we can measure amines using that system. We never saw them in our hourly samples. We never saw them above our limit of detection. Other places where we have measured them, they're usually a factor of maybe like 50 to 500 less abundant than ammonia. So, but they're also more sensitive. So we are always like right around the limit of detection. So we never saw evidence of them. Yeah.
Yeah, I know they would be really efficient with new particle formation.
Yes, definitely. It's certainly possible that definitely in the model, the ammonia might be a surrogate for amines. Because I think their relative efficiency at new particle formation is almost the same as their relative abundance. So that yeah, it's hard to know, but probably in air masses. I mean, that's what we're finding in the chicken farms that we're going to is there's lots of dimethylamine in the air. So if you follow the degradation of uric acid, you can make alkylamines from that as well. Yeah.
I have a question going back to the corn part. So the modeling, if I remember correctly, was on the second year of measurements that you made. Did you do the modeling, like with the stomata and the cuticle and whatever, on the first year? And could you explain that initial spike that you did see in the first year of measurements?
Yeah. So Alex has done the modeling on both years, though I didn't show all of it. But the big spike that we see is like right after the planting. So there's no above ground biomass during that time period. So let's see if I can go back. Approximate yeah. So the big spike we see is over here, and then the plants are like, yeah, I mean,
Okay.
they're proto plants at that point. So that doesn't seem to be that explanation there.
While you're on this slide, I know I see online someone has their hand up, but while you have the slide up, in your modeling, is there a route for other surface uptake in terms of like the plant waxes and things that would be really present in early growth stages? Because I see your model is above your measured. Do you think there's extra loss?
Yeah, we're really struggling to figure out how to manage the cuticular part, because it's possible that so some of the ways that people think that ammonia hangs around on the plant surfaces is through a water film, and they will represent it with a relative humidity dependence. And that doesn't really work for us. And so I think it's possible that just like the bare cuticle has enough adsorption surface for the ammonia that could be responsible. And in separate work that we're doing in urban areas, I'm pretty confident that even like impervious surfaces like sort of glass and concrete buildings and things like that can actually provide a pretty significant reservoir for ammonia as well. So yeah, we're still trying to disentangle what's going on. We have the classic thing in the sort of inverse model where you can create simulations where there's two giant terms that like offset each other because your constraint provides a small net, and then the model decides to make like these bananas offsetting terms, and we're still trying to figure out. I think there's probably a computational approach that will help us solve that.
Let's see that.
I have a question here.
Great.
Can you use hyperspectral differential imaging to measure atmospheric NH3 optically?
Sorry if it's a naive question.
No, it's a great question. I think, I mean, in principle, yes. I don't know. There may be people in the room who know more about whether it's been done before or not. I think the concentrations have to be pretty high to sort of register. But yeah, I think I have talked to people who do sort of like looking at land surface characteristics in the same spectral region or similar spectral regions. And, you know, for them, the absorbing gases are sort of a nuisance, but I think there is a potential here to maybe get some information about the ammonia at the same time. But as far as I know, it's not done on a regular basis from like a drone or anything like that.
Thank you.
Yeah.
I have maybe a small question, and maybe you answered it already. I think you were talking about why you saw so much lower ammonia emissions compared to what some of those previous studies had reported.
Mhm.
And you gave a couple of reasons why. Do you know like what's dominating that? Do you think it's mostly like the physical, like the lower soil temperature, or like the way it's being applied?
That's a great question. We don't really know. I guess in the model, if we believe our bidirectional exchange mechanism, we could, you know, we have control over temperature as a parameter, so we could just say, okay, what would have happened if the soil was that much warmer? But those measurements, the North American measurements that I talked about are, I think they're from the Midwest. I can't imagine the soil was that much warmer, though I should go back and check. So I think a lot of it is down to the management practices, and I think that's why this is quite a tricky nut to crack, because there's not like it's hard to get really granular information about like exactly what, you know, how deep the furrows are and all that kind of stuff about how things are being incorporated. But the temperature is sort of an easy thing to fix, and actually from what I know about many of the inventories, the bottom-up inventories for fertilizer application, the climate impact is usually represented by a sort of binary factor that's either like 1 if you're equatorward of 45 degrees and 0.4 if you're poleward, and that's it, right? It's like so simple. And we know what the temperature dependence is because it's based on physical chemistry. So like it would be in my mind, a very simple improvement to make, and it may not solve all the problems, but I think that would be a good place to start. Yeah.
I want to ask about the instrument characterization figure that you showed with the 2 tiles and the D value. And I wanted to know what drove that D value down through, I don't know if it was throughout the measurement time, or if it was like jumping up and down.
Yeah, so I know you have some experience making ammonia measurements and being tortured by it. I think that variability is mainly from humidity, and also it depends on the concentration itself as well, right? So if you have the way I think about it is like there's a certain number of surface sites in the inlet that the ammonia interacts with, and so if those represent a significant portion of the ammonia that's in the airflow, then you know that's going to be more important. If you have a lot of ammonia in the air, like then that exchange is a smaller fraction of the signal. So it depends on the concentration itself. And then I think the inlet gets dirty through time. And so there's also this sort of like slow increase in D so every so often we would stop our measurements, rinse the inlet with a lot of water, and then start up again, and then it would sort of behave itself better. And as you know, there's another way to approach this, which is to constantly bleed perfluorinated amine into your inlet, which I am somewhat opposed to, since they have like the worst global warming potential of any known molecule. So you have to decide how much the time response of your measurement is worth, but that's a way to solve it. But to be honest, what we found is that if we keep the inlet clean, that perfluorinated amine doesn't offer us much benefit.
Yeah.
If your inlet's really dirty, then it has a huge impact, but if you clean the inlet regularly, it's sort of not that helpful.
And so you're doing that instrument characterization with calibration lines, zero and calibration
Yeah.
steps
Yeah.
throughout your measurement.
Um, Yes, I think like periodically, yeah.
Cool.
Yeah. Any other questions?
Anything else? All right on time. All right, thanks so
Thanks,
much.
Joanne. Thank you.